like to now introduce Dr. Vicky Whittemore. We have a lot of different people with different hats in the room. We have scientists, we have advocates, we have people from funding agencies, and Vicky is all of those things and more wrapped into one. So her formal role is as program director at NINDS, but she has worn all of these various hats and has a really deep understanding from every facet of the questions that we're trying to tackle for SYNGAP1. So we're really pleased she's here to talk to us about how one can think about funding programs that we're all talking about. And I'll just say, as you're getting started, I think Jackie mentioned a lot of things that are seemingly stumbling blocks, and I would encourage us to sort of try to think outside the box about some of those things. Why can't we look at side effects of treatments, and why can't we design experiments that might make more sense than that? And I think that's something to think about as we're talking about NIH funding mechanisms. Maybe drug trials can't take those designs, but if enough good scientific studies follow logical designs, maybe we'll move the needle. So Vicky, welcome.
So thank you, Anne, and it's great to be with you all this afternoon. I'm Vicky Whittemore, and I'm a program director at NINDS, where I oversee grants primarily on the genetic epilepsy, so everything from very basic grants to translational to clinical studies and hopefully clinical trials, as well as grants on SUDEP. And I have a whole portfolio also on imaging. And before I launch into my talk, I just want to go back and answer your question. NINDS is currently funding the development and validation of some wearables for seizure detection. So which would be a device that you put on your head, and so what they're doing now, they know it works, but what they're having to do is validate that what they're collecting from the wearable is the same as what you collect in an EEG. But they're coming, because I think we recognize the definite need for that. So it's coming on the horizon. So how do I advance? Oh, there we go, arrow. So at NINDS, we have 3 divisions. We have a division of neuroscience, which is where I sit in my position at NINDS. And most of the grants that we fund in the Division of Neuroscience are basic studies, very basic studies on synapses and circuits and just how the brain functions, to very disease-focused basic research. We have the Division of Translational Research, which is when there's a finding in basic science that needs to move into validation or biomarker work. I'll talk about this a little bit more. That goes to our Division of Translational Research. And we have what we call the Epilepsy Therapy Screening Program, which is a program where individuals that have treatments can put those treatments through a series of mouse models that is completely free and supported by NINDS. It's a group out at the University of Utah. So if anyone's interested in knowing more about that, just let me know. And then we have our Division of Clinical Research that supports research and oversees grants primarily on clinical trials and pretty significant clinical studies. So I have a number of clinical studies in my portfolio and work on some clinical trials, but really the clinical trials are overseen by program officers in the Division of Clinical Research. So the other day, I took a look at Reporter, which is the program on the NIH website that reports research funding for different diseases, and you can type in anything you want and see what comes up. And what I found was a problem, which I'll have to address, because it says in some years there was 0 funding on SYNGAP1, which I know is not true. But because overall, from 2012 until 2021, there was about $500,000, and on average, dollars in NIH research funding going to SYNGAP1. What I was surprised and pleasantly surprised to see that in 2022, that number is actually 5.6 million, which is really amazing. And I have to say that I think it's in large part due to the funding that you've provided, seed funding that you've provided to researchers that gives them the preliminary data that they need to come in with really strong grant applications to NIH that not only shows the scientific rationale and the need, but shows that the research they're proposing to do is feasible and important and significant. So congrats to you and congrats to the community, and we just hope to see that number grow. Those grants are supported by NINDS, NIMH, and NIDA. And like I said, right now what's primarily being funded is on basic neuroscience research, and we'd love to talk to people and to grow the funding into the translational and clinical research and clinical trial realm. So I was specifically asked to talk about clinical trial funding mechanisms, and we have several different funding mechanisms. And before I sort of talk about some of these, I'll say that what we try to do when a group comes to us with an idea for clinical trial is to say, okay, let us know what you want to do, and we'll try to figure out which is the best mechanism for you. And really work closely with you, and I'll talk about that more on the next slide. But NINDS, several years ago, put in place a clinical trial network called Neuroscience, which is for early phase. So the first phase of trials is phase 1, where it's primarily safety. Phase 2 is where you really begin to look at, is this treatment going to be effective? So those types of trials are being done in Neuroscience. And the way this network works is that NINDS supports the infrastructure and the ability and the personnel to do clinical trials at several sites across the United States. And investigators come into NINDS with the proposal for a trial in Neuroscience utilizing this network, and they can also propose to use additional sites. And that's particularly true for if there's a center that's very specifically working on, say, SYNGAP1, to be added to that Neuroscience network of sites. We also have exploratory clinical trials. Again, these are early phase clinical trials and biomarker studies that really are there to give you the proof of concept that this clinical trial is worthwhile moving forward into a larger, obviously more expensive, phase 3 randomized blinded clinical trial, which are funded through R01 applications. And typically these grants come into NIH as R01s. A lot of time we convert them to what we call U grants. It's sort of a technicality, but that just allows us on the NIH side to really be very involved in oversight and administration of the clinical trial. So one relatively new program at NIH is the ultra-rare gene-based therapy network. And this is a very unique program that sort of is modeled after a program called Blueprint Neurotherapeutics. And the idea here is that if someone has a gene therapy ready to be tested, you bring your idea and submit an application for consideration and review. If it's accepted, then the investigator and their team do what part of the research they can do in their lab, but you're provided with a whole set of consultants to work with you to move that gene therapy forward. And if you need for something to be done that can't be done in your lab, you're provided with CROs, which are companies that will do that particular toxicity or whatever part of the study that is needed to be done that you don't have the capacity to do. And the idea here is to deliver therapeutics to individuals with ultra-rare neurological diseases to really learn from every trial that comes through, every gene therapy that comes through, to standardize and harmonize best practices and protocols to really be able to speed up the process and accelerate the development and discovery and characterization and approval of gene therapies going forward. So at NINDS and across NIH, any grant that's going to come in for review that has a budget over $500,000 a year is required to submit a pre-proposal for review and approval to submit a full grant application. And this is really a process through which we, as the program staff at NIH, can work with the investigative team to really hone the protocol so that when you do get approval to come in for a full clinical trial, you have a pretty good chance of getting funded. And so what we require in the pre-proposal is that the investigative team submits an outline that really outlines the scientific rationale. And this is where having really strong preliminary data from animal models, from cell line studies, is really critical to show that the basis of your idea of why this potential treatment will work is really solid. You propose a protocol, who will be included or excluded from the study, what your outcome measures or your clinical endpoints will be, statistical analyses, and your proposed budget. And there's a lot of discussion that goes on. We usually develop a team. I'm a basic neuroscientist by training. So if someone comes to me with the idea for a clinical trial, I pull in my colleagues from the Division of Clinical Research, and we develop a team to work with the investigators. If it's a device, we bring in people from our translational program that are device biomedical engineers, and we really work very closely with the team to help develop that protocol, to help, like I said, hone it so that when it goes to our internal approval process, it's a solid proposal that you're going to get a yes, you can come in with the full proposal for review. So my words of advice to investigators in the room is contact a program director. So there's actually a whole team of epilepsy program directors at NINDS, and we also work with individuals at other institutes. But the four, I think, key people for SYNGAP1 are two people who are in our clinical division. Adam Hartman's a pediatric neurologist, epileptologist, who is very involved in epilepsy clinical trials and clinical research. Christy Hardy is a relatively new addition to NINDS. She's a pediatric neuropsychologist and is very interested in working with teams to look at outcome measures in all areas of cognition, neurocognition, pediatric neuropsychology, and assessment and methodologies. My colleague George Umana, who's here today with me, he and I both oversee grants on the genetic epilepsies. He takes more of the structure function biophysics grants. I do more of the animal models, characterization, functional studies in animals moving toward clinical trials and clinical therapies. But we're here to help. That's my message to you. And we would love to have people contact us with your ideas. Any way we can help, that's what we're there for. That's our main job, is to provide that customer service to investigators. So with that, I'll stop, and I guess we'll move. I can answer questions now, or we can move to the discussion. So thank you.
Thanks so much, Dr. Whittemore.
Sure.
Recently, I had a similar, customer service experience with FDA officials who referred me to the Bespoke Gene Therapy Consortium, which is a relatively new, I think, sort of hybrid experience between Foundation NIH and NIH. And NINDS is a part of that, and is that something you can expand upon, please?
So I'm aware of it, but I'm not part of that. So I really can't expand on that, but yeah, I'm aware that that exists, that it's a consortium really to help, and it's, I think, helping groups to develop gene therapy where if it's an ultra rare, to move them into the urgent network or to move them into other funding mechanisms. But I'm sorry, I don't have the details on that to answer your question.
Given the mix of folks we have in the room, I wondered if you might speak to any opportunities for academic and industry partnerships through some of these grant mechanisms. Is that possible?
it's possible on all of our grant mechanism, and especially clinical trials, in a lot of the translational studies. I didn't talk about our small business program, the STTR program, which is specifically a program that's a partnership between industry and academia. So yeah, all of those programs are available for those types of partnerships.
So I do have a question. What advice would you give to rare disease organizations where a gene has just been newly identified and linked to a phenotype, and there aren't a lot of researchers in the space? And it's a lot of times, a lot of these, patient advocacy organizations are started by champion families. Microlia is, a great example. But what advice would you give to patient advocacy organizations in starting conversations with the NIH and trying to recruit researchers into the area for the gene?
No, that's a great question. So just by way of background, I spent many years working with the Tuberous Sclerosis Alliance. It's an organization that was founded in 1974. My nephew was diagnosed when he started having infantile spasms when he was 3 months old in 1985. So I joined the board in 1986. I was a young junior faculty at the University of Miami, but determined to make a difference. And they were, at that point, giving out $1,000 here, $1,000 there to their favorite doctor to do some research. So we put a robust research program in place. And so I think that's the first thing you need to do. But in terms of contacting NIH, where we can be helpful and where I've been able to help in young and new groups, is we can go in and we can search the database and say, who's ever submitted a grant on that gene? And so, for example, an example is hypothalamic hamartoma is a type of epilepsy where there's mutations in just a very specific part of the brain where they know that those cells have mutations in a gene called sonic hedgehog. So I was able to go in the database, pull out all the researchers that had ever done anything on sonic hedgehog, which was in mostly all basic science, give that list of investigators to the organization who they contacted, and now they have a whole cadre of researchers working on that genetic disease. And so that's the way that we can be helpful in terms of helping to guide you into people that are potentially already working in your area or in a related area. The other thing is, in the Tuberous Sclerosis Alliance, we were actually quite fortunate once the genes were discovered in the 90s that the researchers figured out that the genes were in a very important signaling pathway, the mTOR pathway that's important in cancer. So we very strategically hunted out the cancer labs that were doing research on that signaling pathway, supported a postdoc in that grant. So now you had a postdoc studying what those genes were doing in TS in epilepsy, surrounded by all these people who were studying that signaling pathway that just catapulted the research. So thinking quite strategically can really help your research efforts, and I'm happy to have an individual conversation with you or any group that's interested in having those kinds of conversations, because like Ann said, I've been there, done that. So happy to have conversations with anyone about those. Okay, thank you.